My name is Jelani, neuroimmunologist by training, and I'm one of the executive leads at DeSciWorld. And very briefly, DeSciWorld as a platform's whole focus is on broadening the awareness and participation in decentralized science. So providing easily digestible format of information so that people can learn about some of the amazing projects like the BioDAOs that we just saw, and many of the other projects that we're going to see today, and learning about and how can they participate, how can they be active, and how can they help push the mission statement. So that's what we're here for, and that's what we like to do. I'm going to be the moderator for the next series of talks and panels, and I'm looking very much forward, and I'm very thankful to the ResearchHub Foundation and SciCon for giving the opportunity to do so. So with all of that being said, I'd like to kick off this by first introducing Dr. Niklas Rindtorff here, who is the co-founder and CEO of the Open Lab Association, which is alternatively known as LabDAO. Dr. Rindtorff hails from the healthcare industry, where as a physician scientist, his research focused on improving treatment recommendations in specific oncology through image-based profiling of compound effects in patient-derived organoids. And that's something that's very close to my heart as somebody who has worked in the development of stem cell pipelines and understands the need for these somewhat in vivo, in vitro hybrid situations that can help us better understand treatments. Now, this commitment to improving quality of life through science has since followed him in his journey into Web3, where he has helped co-found LabDAO, an online research collective that provides access to and develops open tools and infrastructure for computational life science research. And in today's talk, he's going to be regaling us and giving us the pleasure of learning about one of their flagship tools, which is known as Plex. And I'm very much looking forward to it. And I'm sure I can speak to the rest of the crowd for that. And so with all that being said, and without further ado, Niklas, it is very good to see you again. Thank you for sharing your time with us and giving us the opportunity to learn about some of the great things that you guys are doing at LabDAO and how we as scientists and science enthusiasts can leverage Plex to help improve our research. So the floor is yours.
Thank you so much for the introduction, and thank you to the whole ResearchHub team for having me on this great conference. Yeah, as Jelani already introduced, my journey started very much in translational research and in more particular in image-based profiling, which is, for the uninitiated, a very data-intensive way to study how cells or any form of in vitro model of disease reacts to treatments. And during these projects, both in the States, but also in Germany, where I'm calling in from today, the recurring theme that we always ran into was that we accumulated lots of data. And I'm speaking not sort of genomic scale data, but really, it's a lot of imaging data. So we're easily talking terabytes per experiment. And over the course of, a couple of months, one could easily accumulate hundreds of terabytes of information. So very quickly, this type of research, although it's sort of biomedically motivated, is very much determined by your ability as a scientist to process large data and to then also effectively share large data. And this was very much sort of the origin story for LabDAO, this idea that there should be a way for scientists to share their data and the tools that they also build with each other in a more permissionless and direct way. And for that, for the next 30 minutes, I'm going to be also able to share some slides. So the initial idea that we had with the Lab Exchange was that we wanted to create a space where people can trade laboratory services. So usually when you were in a university, you would have something like a core facility. But what is the core facility for scientists that are living in countries that might not have all the infrastructure that they need to get their work done? Or where's the core facility for startup founders that are not based in any of the large hubs? How can they sort of test an idea? How can a college student, for example, run an experiment if they're able to convince enough people online that their idea has merit? So with this idea, we said, okay, we want to create a new form of organization, a DAO, around this idea of a lab exchange, where there would be permissionless participation, distributed storage to make sure that whatever data was created was kept in a secure way, and also token-based ownership of the results, so that if I was located in location A, I could pay someone in location B to process data that I want to be analyzed, and tokens can track who has rights to what pieces of the data that is being created, and there's a neutral platform on which I can pay that scientist to do the work. So with this open source exchange outlined, what we, however, relatively quickly understood was that this was a very large task, and we needed to break it down. And when we looked at the state of computational biology, it was clear that there was a lot of room for improvement. If this was sort of an in-person event, I would ask the audience, like, what you think you're seeing here. But I'm just gonna, like, after you can take a moment of looking at this, I can explain to you what's going on here. So this is an example from a tweet in a laboratory, actually in a relatively well-equipped laboratory in Munich, where in order to run a computational biology tool that was very popular at the time, and I think still is, the scientists went back to using these free Google Colab notebooks. And because Google Colab notebooks, they time out. They put the mouse of the computer on a roller to keep the mouse active, so the computer doesn't time out and the Google Colab notebook session doesn't time out. Why is this sort of, you know, the way that we still share computational tools between scientists, using sort of that type of platform where scientists can't just permissionlessly, for example, use the tool? They cannot scale it up easily if they need to, and they cannot track results because these types of notebook sessions, they die out extremely quickly. So what we did was we sort of simplified the spec of the Lab Exchange in its first version, and we called it Plex, where Plex is the Plex Lab Exchange. It's a Python package that enables permissionless participation, distributed storage of results, and distributed compute as well. And I'm going to go into a bit of what all of that means in a bit. So when a user is accessing the Lab Exchange, they're submitting a compute request, which can basically be any arbitrary Docker container for all the computational biologists in the audience. So those tools can be, you know, protein folding, small molecule docking, protein designs. These are applications that we already have. Applications that we're onboarding in the next couple of weeks are going to be genome assembly, genome annotation, and then also I think some more sort of exotic applications that are related to drug discovery and sort of more advanced drug discovery. Now, one thing that, however, was missing in the Lab Exchange until recently was token ownership. So when I submitted a request to the compute network, one of the nodes in the network would pick that request up, and they would process it, and they would post the results back on a distributed storage system that's called IPFS. I'm going to go into that in just a bit. But there was no sort of permanent record of that work being done. And the person that submitted the request to the exchange, they didn't keep something. They couldn't claim that they actually did this analysis run. And also they couldn't share it easily what they just did until recently. So what I'm introducing today, and we announced it, I think, a couple of days ago, is records, and something we've been working on over the last weeks after we launched the Lab Exchange earlier in the year, which allows you ownership, grants you ownership rights over the scientific data that you create. So it's, again, sort of integrated in our fully open source Python API, and gives you access to some of the latest biomachine learning tools running on top of this distributed compute and storage network. Every time you mint a record, you're assigned rights to the output, so that those are just basic copyrights right now to all the creative output that might come with the work that you've done. But what we're really interested in is eventually bundling these records into like fully fledged intellectual property claims that, you know, are equivalent to like a patent application. The way that you can use this right now is literally from any place where you can run Python. So this is just an example out of a Colab notebook. And when you use Plex and you want to mint a record, you know, you can, for example, install the package here, and you want to run AlphaFold. You can put in the sequence of the protein where the structure is that you want to predict. You put in your wallet, which is basically your user account, and you hit go, and off, you know, you sort of send out a compute job, and eventually when everything goes well, you do mint a record. Now, how does all of this work? So I'm going to walk a bit through, you know, the three pieces that, you know, records consist of. At the bottom layer, we have a storage system, and on top of that, we have a compute system that's called Bacalhau. And in the top layer, we have a permission and ownership system that's just powered by basic NFT contracts that we're running on Optimus and testnet at this moment in time. some of these words might not be familiar to everybody in this audience. So I'm going to take some extra time to explain to you what's behind these individual technologies and why I believe they're so ridiculously important to the way that science as is going to work. I think generally when we talk about decentralized science, we think a lot about levels of ownership and engaging online communities, but I do seriously believe that, once you come for that type of excitement and the community engagement, but I think you're eventually going to stay for the sort of fundamentally superior guarantees that some of these, compute infrastructures give scientists to collaborate more easily with each other. I'm very excited to go into them now step by step. So at the bottom layer, we're looking at storage. And, when you have files on your computer at home, most of these files are probably addressed with, home/Niklas downloads project 1. And that's where you keep all your files. And if you build a script, a piece of code that analyzes this data, and you share the script with someone else, that's most likely not going to work because their first name is not Niklas. They have a different name. And so their laptop has different file system structures. So suddenly whatever you did is not reproducible. And now people have worked on, solving these problems, and there are multiple different solutions. But I think one of the most elegant ones is content address storage. So here you use a hash function to create unique identifiers for every data file that is available on the storage system. And we're using one of these storage systems that's called IPFS, which is the largest of these distributed storage systems. And what this address gives you is it's a unique hash identifier that has standard length that is the unique address of this file. And no matter where in a peer-to-peer network that file is located, you will always find that file based on this address. And this is obviously a more robust solution relative to a standard server client model that we're knowing from the sort of Web 2 world, where you just need to hope that the laboratory or department server doesn't shut off or if you still want to keep access to your data. And there's probably always one PhD student that has the very unfortunate role of making sure that this department server keeps running depending on where you are in the world and what the infrastructure of your research organization is. So this is a more robust sort of way of storing data. Now, what's even more interesting, I believe, is that you can pair these distributed peer-to-peer storage systems with also peer-to-peer computing systems. And the reason why I'm excited about this, because there are two problems in biology that are very prevalent, and peer-to-peer compute does solve both of them. So the first problem is we have very large data sets depending on where we are. And then the second problem is we always have very prototype grade code with very exotic dependencies. So we have serious data, and you could say for anybody who's worked sort of in tech professionally, pretty unserious code. And so the solution to make both of these work with each other is to move the compute into containers. So whatever code we're writing, we just try to bottle it up so it is as robust as possible, and it doesn't matter on where it is run. And then because the data is very large, we don't move the data to the compute, we move the compute to the data. And this is what the Bacalhau network enables. And then, on the third layer, not only do you need to have a sort of container runtime, so basically a system that allows you to run arbitrary containers, you also want to perhaps, add your own tool, add your own container, and make that as easily as possible. So what we looked into was sort of inspired by a standard that's called the Common Workflow Language that allows you to specify arbitrary, applications by you just filling out a JSON document. So if you, for example, have a tool in your laboratory and you're counting cells, and you have a container in which that code exists, you can put that into one of these documents, and you say, okay, the input is for example, a file that is an image, and the output is a CSV file that has all the cell counts. And then you give the command line argument that you would run, and it would then be a tool that can already be executed on this peer-to-peer network. The philosophy that we follow when we design these types of specifications is very much sort of inspired by sort of consistent instructions that we know from like recipes. So here, for example, we have, when you want to create a pie, you have very clear definitions of what are your ingredients. For everybody who's interested in category theory, you might remember this sort of meringue pie example that I'm showing you here. Then you need to have a block in which you define, what's actually happening under the hood with these ingredients. And then you need to type your output. You need to be specific about what comes out. And we took this approach, like all the applications that we're onboarding, where, for example, when you run a docking model, you have a protein with a very specifically typed file that is that protein, and then you have a typed input for the small molecule. You have a well-described set of operations that are happening under the hood, and then eventually you end up with an output that is well-defined. Now, we have, as I said in the beginning, we've onboarded a whole series of tools. So we have tools for small molecule binding, protein folding, and there are more tools coming as well. You can head to our docs.lab.xyz to learn about what's out there. And then, as I said, there are also more tools on the way: genome assembly, protein binder design. So actually designing proteins that bind tightly to a target that's implicated in the disease or that you want to further study. All of these tools are already available, and we would love to help you use them in whatever context your research might find them useful. Now, the third layer of the stack are non-fungible tokens. So this is literally an example on OpenSea testnet that you can look at yourself today. So all our records, they have sort of random generated names right now. And when you look under the hood of what these tokens contain, I think all of what I just told you comes together really nicely. So every token that we mint basically has a section that we call sort of a graph that contains the specification of the input, the specification of the output, and a tool reference that makes sure, that contains the container image and all the input-output relationships. Now, in this particular case, we docked a small molecule against a protein. This is a very standard thing that you do in the earliest stages of a drug discovery project. And we have these unique hashes that I mentioned in the beginning for both the file that is the protein and the file that is the small molecule, and the same thing also for the thing that came out. So with this, we believe we have basically the most reproducible format to capture what a scientist did. In this particular operation, the scientist took two files, they docked them against each other, and they have a record of doing that at a given moment in time, because all of this information is minted as an NFT on chain. And because that is not sort of, I believe, cool enough that you can actually show others, this is literally what I did, and you can rerun it, and we can work together, and you can, build on top of my Lego block. What we also do is we dip into certain license contracts that have been developed, for example, by the A16Z crypto team. So all of our contracts right now, all our smart contracts that you use to mint these NFTs, they give the holder of the NFT an exclusive license on the copyright and data ownership rights. And again, as I said in the beginning, what we're really interested in is building the infrastructure that now lets you collect all these Lego blocks and eventually bundle them together into serious intellectual property portfolios, so that you as a scientist can collaborate with whoever you want, and you can sort of start, really collecting the work that you're doing. Now, these licenses also let you choose the terms under which you want to license your work. So if you, for example, don't want to have an exclusive license to your work, you can also do sort of a CC0 style license to your work. So you have the flexibility to just control how your work should be treated. What can records do? So we're using this technology pretty actively and trying to really test it in the field. So what we were looking at is partnering with companies and also academic researchers that have a computational biology problem. They think one of our tools might be helpful, or they have one particular tool in mind that they would, like us to onboard. We are currently looking for these types of pilot projects, and if you're interested in working together, we would love to, get to know you. Here's some examples of what we're doing right now. This is a pilot that we're doing with a company out of the US, Leo Labs. They're designing protein binders for oncology targets, where we're using some of our protein binder design tools, Colab Design, RF Diffusion, Protein MPNN, to design proteins that bind very tightly to surface markers that are associated with cancer cells. Here's another example of a company that we started working with. And this is an engine, a docking engine. It's called UniDock. It's extremely fast on A100 GPUs, unprecedented, I think, speed, or definitely sort of in the top in the top tier of high velocity docking engines. And here, we onboarded these tools and we make them available, and we test, to what extent are they competitive with other platforms that don't have a lot of these guarantees that our system has. And finally, here's an example of a collaboration with another DAO where we analyze data for this community and help them sort of make decisions in their further drug development sponsoring process. Last, I think one sort of relatively recent case study that we did was we partnered with Protocol Labs to move the whole Facebook AI research protein structure catalog, the ESM atlas, onto IPFS, and hopefully soon also Filecoin, so that this data is preserved and also right at the fingertips for scientists that want to use our platform to do their work. Now, these might be a bit abstract because they're basically, these vignettes are telling you what we're doing for other people. If you just want to get started today, here's some things that you can do right now. We launched about 30 days ago, and up to this point, about 200 records have been minted. And we would love you to also mint one. We have new tools that we just ordered, for example, this protein binder design tool. And you can head to try.labdao.xyz. I'm going to share that link in just the end of the presentation to give it a spin and mint your own. Now I want to sort of end, and I know I'm also coming up on time. I want to end with what I think is sort of an ecosystem view. How do we at LabDAO, how do we at Convexity Labs, the company that's supporting the project, and the Open Lab Association that's hosting it, how do we think about the components of this ecosystem, this wider ecosystem, and where we fit in? And what we currently see are two components. So we have governance tokens and we have legal sort of wrapper tokens. So governance tokens, these are tokens, ERC20 tokens. I think ResearchHub has a governance token. A lot of these other bio-DAOs have governance tokens. And these tokens can be used to, for example, pool funds to sponsor research. And this is sort of the perfect segue to this other token type, which are these legal wrapper tokens. So these are tokens that you mint in collaboration with a university tech transfer office, where the online community has pooled their money and they're now paying the university researcher in return for some legal guarantees to the intellectual property that is handled by the technology transfer office. So usually there's basically an edge between the governance token and the legal token. You raise funding through the governance tokens and you decide on what to fund, and then in return for funding a scientist, you get a legal right. What is sort of missing from our perspective is the actual direct relationship with the scientists, and especially this sort of bottom-up scientific relationship. So this is very much sort of a Netflix model where you bring together your online community and then you partner with movie studios and you sort of out-license the movies that they're producing. We're way more interested in YouTube than in Netflix. We want people to just create and not have to ask anybody. And we want people to just create, collaborate with their creation and see where it takes them. And maybe their creations are the starting point for their own DAO, where they're interested in one particular aspect of scientific computing, or what they start accumulating might eventually lead to a legal contract, or might fit really well with an existing sponsored and financed research project. And this is really what we want records to be, sort of closing the loop of, I think, the decentralized science and decentralized finance stack, where now scientists themselves have their own cultural product and they can contribute it to the ecosystem, where I don't need to have my own online community and I don't need to have a sponsored legal agreement to do my research. I just get going and I start collecting these records, and I see where it takes me. Maybe I find other scientists and I vibe with them and I start creating my own online community around the records that we've amassed. Or, eventually, perhaps, I can take this a step further and turn it into a fully financed and sponsored research project. Now, with that, I hope I was able to give you an overview about the Lab Exchange, what you can do right now, how records fit into the equation, and our current perspective on this wider ecosystem that we sort of continue to be extremely excited about. Thank you. You can take a look.
Amazing. Thank you.
Of course. you can take a look at try.lab.xyz to run a notebook in a couple of steps, put it in your wallet if you want to keep the work that you've done. And otherwise, we'd love to hear your feedback.
Absolutely. I think Plex is an amazing tool that pulls together. And I like the idea of bridging that divide between the crowd community, so their ability to raise funds, the tech transfer, which is very important when you have scientists living in the academic sphere, but also involving those scientists as well in that whole process. That trilemma is actually very, very important. And so, as Niklas said, we'd like to open up the floor for people who may have questions. So feel free to put those in the question and answer or into the chat, and we'll field them from there. But one of the questions that I have is kind of to that triumvirate point. Have you had the opportunity to speak with university institutions in terms of onboarding that? So we know that there is friction as it relates to who owns what, how that works within the current framework of institutions across the world. And what have those conversations kind of been like with you?
Yeah, so our focus has mostly been always directly with the practitioners. What we built, the protocol we built has been very focused on practitioners. So we spent most of our time talking to scientists that actually have a computational biology problem, and we try to help their computational biology problem first. Now, I think as we keep doing that, and as we onboard tools that computational biologists or other scientists that actually, ideally, scientists that are not very familiar with computational biology, they just want that one thing to work. As we sort of build a product that increasingly brings value to them, we definitely do have to have these conversations with universities too about what these tokens mean. And I'm frankly pretty excited to start having these conversations. I know there are some pilots, for example, in the Max Planck organization with, they call it Blocksberg, where they keep track of scientific work that's being done, and people can register that. But I also think that other university systems would be open to giving their scientists a way to record the work that they do.
And in your experience, since you're dealing with really that ground community of scientists, what kind of approaches have you guys taken to really get involved, to get plugged into those individuals? Now, clearly this is one where you can help broadcast, but what are some of the others?
So it's really been, what we see on Twitter and Discord, and then we have conversations just with computational biologists, sometimes PhD students, sometimes professors, sometimes founders. Actually, we really like working with biotech companies because they just really test. They have no patience for something not working. So we really, pressure test what we're building. And then I think, however, as a next step, we're really interested in also talking to universities directly. But it's mostly people just through the community contacting us, talking about a project, and it's something that we really enjoy. And I think that's sort of obviously a benefit of having an active online community like LabDAO and a lot of other communities too.
For sure. And I know you guys have your weekly LabDAO's Twitter spaces that are hosted by Jocelyn and Stanley, and a few other people are there. So that's also a great opportunity for those listening. If you want to get involved and start to talk with the founders and the rest of the community at LabDAO, you should definitely join those. I think they call coffee, they're like coffee meetups, and those are usually fun. To your last point, I think this will be the last question until we bring up our next speaker, but you talk about biotech and the application of this in that framework. Now, that's an enterprise-based community or enterprise-based entity. I assume that the considerations are different between that and those non-enterprise-based entities. And so what are some of the amendments or considerations that you guys or LabDAO has to put into play when looking to deal with those entities that require more level of privacy, more legal sophistication when it comes to this?
Yeah, it's a really good question. One aspect to the exchange that I haven't talked about in this presentation, but that we've been relatively busy with, is the option to basically only broadcast the work that you did through these tokens, but not necessarily use the fully public IPFS system and public compute system, but basically create your own community workspace or your community internal or company internal compute and storage environment. And this gives, I think, both companies, but we hope also these like BioDAOs, the ability to have their own intranet and make sure that they can actually innovate. And while we might, and I'm definitely on that list where we might not find that particularly thrilling, as sort of, in the context of open science, that I'd rather have that type of like internal science happen on rails that can very easily, with the flick of a button, be fully distributed, A, and then B, we also just need to understand the sort of realities of therapeutic development and what it takes for a scientific idea to turn into a medicine, and being able to like claim intellectual property over your work, is part of that. So we need to build products that respect that reality if we want to really bring like a change.
Yeah, for sure. I think Laura puts it very nicely in the last panel, where we have to go to where we're seeking to. So we do want the enterprise grade because they do form there's a value chain that exists there, and how do we helpfully kind of bridge that divide? And that's going to be a constant conversation and a dynamic change in the policies and the infrastructures that we are making on the ground floor, and coordinating with them to see what best suits them. But it's essential and it's necessary. The point is to disrupt, but not necessarily get rid of what existed before. So with that, we don't have any more questions. Niklas, as always, it is a pleasure to hear you, your passion and what you're doing. Thank you very much for the great things. And as a scientist, I appreciate the protein docking and all the biological computational aspects. I tell all my friends about some of these great platforms. So thank you again. Have a great evening. And thanks for coming on.
Thank you for having me.
Absolute pleasure.